Hello, everyone. My name is He Xu from Professor Nai Hejin's lab. I'm delighted to announce that our research article titled Generation of Functional Posterior Spinal Motor Neurons from HPSC-derived human spinal cord neuroprogenitor cells has recently been published in Cell Regeneration. I would like to express my sincere gratitude to the editorial board for granting us this wonderful opportunity to present our work. Well, let's talk about neurons. Neurons come in many sizes and types, but a motor neuron controls our muscle contractions. They can do this by receiving messages from the spinal cord and sending them to the muscles that are required to complete the desired action. Well, motor neuron disease, such as ALS, causes a neuron to gradually deteriorate and eventually die, affecting its ability to transfer messages from the spinal cord to the muscle. Well, as a result, this decreases the body's ability to control a muscle or contract it. In 2019, there were over 1 million individuals had the motor neuron disease, and the prognosis is only 3 to 5 years. What's worse, currently there is no cure, and we lack efficient disease model for drug discovery. Human-induced pluripotent stem cell, short for HIPSCs, have demonstrated great potential for studying motor neuron disease. Like we can say, somatic cells from ALS patients can be reprogrammed into iPS, which can then be differentiated into motor neurons. These motor neurons retain the genetic background of the patient and can be utilized to mechanistic studies and high-throughput drug screening. Scientists worldwide have developed numerous motor neuron differentiation protocols, which have been shown promise and been widely used. However, these protocols were often long, expensive, and inefficient. Another devastating disorder is spinal cord injury, where stem cell therapy holds high promise. Stem cell-derived neuroprogenitors combined with scaffolds and growth factors can be injected into injury sites, contributing significantly to the recovery of neural circuits and ultimately restoring motor function. Scientists have attempted transplantation of HPSC-derived neural stem cells into rat model of spinal cord injury. Staining results showed excellent survival of the grafted cells in vivo, with long axon extensions, even observed in the olfactory bulb. However, behavioral tests didn't show functional recovery. When examining the detailed marker expression of the neuroprogenitor cells derived from either rostralized or caudalized iPS or H9 cells, it was found they could only express forebrain, midbrain, and hindbrain markers, lacking the expression of specific markers for the spinal cord, like HOXC6, HOXD9, and HOXC10. The HOX gene family consists of 4 clusters and in total of 39 genes. The higher the number, the more posterior the original identities. We can say HOX 4 to 8 are expressed in the cervical and brachial region, and HOX 9 are expressed in the thoracic region, and HOX 9 to 11 are expressed in the lumbar section. Several studies have already shown efficient differentiation of anterior spinal motor neurons from iPS. However, generating high efficient posterior spinal motor neurons from iPS remains challenging. In 2015, a hypothesis proposed that the dual origin of neuroprogenitors based on studies in chick and mouse embryos. The posterior spinal cord and presomatic mesoderm both developed from a bipotent progenitor known as neuromuscular progenitor, short for NMP. During mouse embryo development, NMP are located near the node region at E7.5, and by E8.5, they differentiated into neural and mesodermal progenitors and migrate to form the neural tube and zomite. Based on this hypothesis, we ask the question of how to generate functional posterior spinal motor neurons from HPSA-derived NMPs. We designed the following workflows: differentiating IPS into NMP and patterning them into spinal cord neuroprogenitor cells, short for SAMPC, and then directly differentiating the SAMPC into spinal cord motor neurons. We can say after 4 days differentiation, NMP were generated from pluripotent stem cell. An immunostaining of NMP markers such as SOX2 and BRCA-T, the double positive cell population representing NMPs revealed a 90% high efficiency. The high purity NMP were then patterned into neuroprogenitors, which could be passaged in vitro up to 40 times, providing sufficient cells for clinical applications. These NPC were characterized by their expression of pan-NPC markers, including nestin, SOX1, SOX2, and PAC6, which showed homogeneous expression. The proliferative marker KX67 also exhibited high expression. Additionally, the NPC expressed the spinal cord specific markers such as NKX6.1, NKX2.2, and OLIC2. And the RNA sequencing results of SNPC at different passages demonstrated a high correlation with developing human spinal cord rather than the developing brain, indicating that our NPC are spinal cord specific neuroprogenitor cells. Next, we directly differentiate SNPC into motor neuron. The SNPC could differentiate into motor neurons as quick as day 6 in vitro, and further maturation were observed during extended culture. These motor neurons exhibited a high expression of motor neuron markers such as HB9 and ILIT1. They also demonstrated homogeneous expression of mature neuronal markers, SMI32, NeuN, and MAP2. By characterizing the neuronal subtypes, we stained for different neurotransmitters, including chat, WeChat, WeGlut, GAT67, and TH. As we expected, these neurons only specifically expressed cholinergic neuronal transmitter markers, chat and WeChat. Furthermore, we characterized the expression of Hox proteins through immunostaining, which showed that our motor neurons specifically expressed posterior markers such as HOXC9 and HOXD11. Functional maturation of HPSA-derived neurons often take a significant amount of time in vitro. To assess the functional properties of our spinal motor neurons, we performed patch clamp recordings during a long-term culture in vitro. Action potential signals were detected as early as day 6, and the percentage of cells capable of firing action potentials reached 100% after day 24. According to these results, we can see the window of functional maturation range from day 18 to day 42. We also utilized a high-density MEA system to characterize motor neuron function in a high-throughput manner. Consistent with the patch clamp results, MEA recordings also indicate the functional property of spinal motor neurons from day 18 to day 42. Additionally, we performed axon tracking, which revealed an increasing abundance of functional axon over time. Finally, we developed a stable co-culture system of SAMPC-derived spinal motor neurons, spheroids, and C2C12-derived mouse skeletal muscles. allowing us to visualize NMJ-like structure in vitro. In summary, we successfully generated human spinal cord neuroprogenitor cells from HPLC-derived high-purity NMPs. These SNPC can be efficiently differentiated into posterior spinal motor neurons, which exhibited fast electrophysiological maturity in vitro. Furthermore, the co-culture of spinal motor neurons fluids and C2C12-derived muscle fibers resulted in the formation of neuromuscular junction-like structures in vitro. I would like to express my heartfelt appreciation to all the collaborators and mentors who have contributed to this research. I'm deeply grateful to Yao Yao, Fenyong Yao, Jiehui Chen, Meishi Li, Xianfa Yang, Shen Li, Fangru Lu, and Professor Naihe Jing, Professor Guangdeng Peng, Professor Shuijing He, Professor Ping Hu, and all the S308 members. Thank you all for your attention.